Hello, my name is LeeAnn Hewitt, and I'm here to present my poster on a novel dorsal root ganglia protocol. I'd like to thank my mentors within the Diabetes Institute, Dr. Campbell Thompson and Dr. Scarden. Let's go ahead and get started. So as we all know, diabetic neuropathy is one of the most common complications from diabetes, which affects over 10% of the US population. Dorsal root ganglia, or DRG for short, are pseudo-unipolar sensory neurons that transmit information from crucial endocrine organs and peripheral extremities to the spinal cord. Given their location, there's a promising possibility that these ganglion can play a major role in reversing diabetes-induced neuropathies. However, technically challenging DRG dissection and cell culture protocols are often difficult to replicate. The purpose of this study is to determine a simplified protocol to dissect and isolate mouse DRG cells and observe if those cells achieve neuronal outgrowth. So methods. So 5 lumbar DRG were dissected and extracted from a female mouse using iCook protocols. 2 major points specific to this protocol include that the animal was kept in a prone position to ensure a constant point of reference throughout the dissection, as well as keeping the spinal cord intact as a dissection landmark. So you can see here that these bulbs represent the DRG, and as well as this going down on both sides is a sympathetic chain, which can also be used as a dissection landmark. So for the primary culture procedures, 1, the DRG were placed in PBS on ice and washed in culture medium. 2, the DRG were chemically and mechanically dissociated using trypsin and light vortexing. 3, a live cell count was performed using a trypan blue and a hemocytometer. 4, the cells were washed in fetal bovine, plated, and placed in the incubator. 5, the primary culture was maintained throughout the lifespan of the cells. And you can see on the opposite side the modified reagents that were used and specific to this protocol that really are more accessible and really do help with the replicability of this particular protocol. So for findings, the simplified isolation technique was successful in producing 300,000 live cells. Upon plating, most of the neurons were slightly swollen and did not attach overnight despite the culture plate and flask being plated with poly-L lysine. However, a cluster of isolated cells did attach and developed really consistent neuronal outgrowth, and that culture was maintained for 15 days. And you can see here throughout the timeline that this initial plating, they were a bit more bulbous than the neurons I've worked with in the past. And then you can see here that there's elongation and outgrowth, and it really does take stride around the 6th. But really this meshwork and beautiful healthy cells really developed on the 12th. And this was the largest outgrowth noted throughout the experiment timeline. So a little discussion. The live cell pellet and neuronal outgrowth produced indicate that a simplified protocol is possible. These live cells really can be effectively extracted without removing the spinal cord, replacing the dissociation medium with PBS, using more accessible reagents, and using both mechanical and chemical dissociation. This protocol indicates a more accessible method can serve to simulate a human diabetes disease model in mice exposed to STZ or streptozotocin, and that future studies really should address if the swollen state of the neurons could be attributed to a pH imbalance, osmotic pressure, or indications of cellular injury. The relevance to nursing. This model, if replicated and validated, would indicate a viable translational bench-to-bedside model to test diabetic neuropathy reversal treatments. This protocol really does set the stage for increased DRG culture accessibility and future treatment discovery that will improve patient health outcomes. Thank you so much for your time and attention, and I really do look forward to answering any questions you may have. Thank you.